Hello, my name is Saul Laredo, and in this video I'll be explaining the membrane structure and functions. Okay, so divided into two sections, structure and functions. The plasma membrane contains both structure and has structure and function. I'll be going on from the plasma membrane to the phospholipid bilayer, which is the most single basic unit that makes up the membrane. The phospholipid bilayer contains a hydrophilic head and a hydrophobic tail. And an amphipathic molecule is a molecule that contains both hydrophilic and hydrophobic parts. Detergents are exactly that. They are an amphipathic molecule. They have both hydrophilic and hydrophobic parts. Here I'll go on to membranes. You have integral membrane protein, peripheral membrane protein, and proteoglycan. The integral membrane protein is a protein that is directly attached to the membrane. The peripheral membrane protein are not attached to the membranes. And the proteoglycan are proteins and membranes. They have one or more polysaccharide chains. Moving on to our fatty acids, our saturated fatty acids, and unsaturated. Saturated is a fatty acid tail with no double bond, and unsaturated does contain a double bond. I'll be moving on to the solutions. Hypotonic, hypertonic, and isotonic. Hypotonic solution is a solution that contains more water than solute, which then we have hypertonic, that's the opposite, containing more solute than water, and isotonic is an equal balance of solute and water. Here we have our models. We have our sandwich model, Dapson and Danieli, also known as the Dapson-Danieli model. This sandwich model is one of the first models that was shown to us explaining the phospholipid bilayer. And the the fluid mosaic model, also known as Singer and Nicholson model, is the most accurate and the most used chart that we use today to explain how the phospholipid bilayer looks and interacts. Here we have our associated proteins. The monolayer associated protein would be a membrane protein that's anchored to a cytosolic surface by an amphipathic alpha helix. And here we have our lipid-linked membrane associated protein, either attached to either side of the bilayer solely by covalent bond. And the protein attached membrane associated protein is these proteins are attached to the membrane by a weak non-covalent bond. And here we have our covalent bonds. We have glycolipid and glycoprotein. Both are very similar. The only difference would be that a small oligosaccharide bonded with a lipid, and this would be a small oligosaccharide combined with a protein. I'll be moving on to the function side. I'll start over here with the gradients. An electrochemical gradient is the net driving force. This is what determines if the direction of passing transport across the membrane. And the concentrating gradient is the process of particles moving through a solution or a gas. I'll go here to the channels. Here we have a channel. I put the channel mediated passive transport, the membrane channels, gated ion channel, and an ion channel. The gated ion channel is a response to different type of stimuli depending on the type of ion channel. We also have our membrane channel, which is the small hydrophilic pores in the membrane which solutes pass by diffusion. And the ion channel would be the same thing. The only difference is the ion channel lets ions pass through. Here we have our membrane. We have our transporter membrane, anchor membrane proteins, receptor, and enzyme membrane proteins. The transporter membranes, these are the ones that decide what molecules and membranes will pass through. The anchor membrane proteins, these are attached to a matrix. The receptor membrane proteins, these receive chemical signals and they transfer it to the molecule, to the membrane. And the enzyme membrane proteins, these make the catalyze what happens within the membrane. And we have movement, we have simple diffusion, and osmosis, vesicle, and I'll be explaining these first. Simple diffusion is when a molecule can pass through the lipid bilayer with no aid at all. Osmosis would be a change of when water transfers from a high water concentration to a low water concentration. Vesicles allow membranes to move from one part of the cell to another. And here we have transport. We have passive transport and active transport. Passive transport requires no energy for the transport to occur, and active transport does require energy. And I believe the last two parts for me to explain will be selective permeability. This would be the membrane. Some molecules and some things are allowed to pass through the membranes and some are not. This is allowed through selective permeability. And sterols would be the carbohydrates found in animal cells. And this would be my chart of membrane structure and function. Thank you.